The overall goal of this methodology is to achieve highly efficient transduction of primary human bronchial epithelial cells using lentiviruses.
This method can help researchers who want to use primary human bronchial epithelial cells rather than cell lines and might be adaptable to other difficult-to-transduce cell types. The main advantage of this technique is a high transduction efficiency maintained throughout differentiation into a pseudostratified epithelium.
The protocol should be executed in line with institutional and governmental regulations under enhanced BSL-2 conditions in the USA. After preparing HEC medium in a collagen-1 coated dish according to the text protocol, use 10 milliliters of the medium to plate 1 million HEC cells onto the coated dish. Incubate the dish at 37 degrees Celsius and 5% carbon dioxide until the cells are 50 to 60% confluent. This will be considered day 0. On day 0, in a laminar flow cabinet using a commercial transfection kit, bring reagents to room temperature. For each 10 centimeter dish of HEC 293 cells, in a 15 milliliter centrifuge tube, combine the following: 4.5 micrograms of PMD2 VSVG envelope DNA, 7.5 micrograms of PMDLG PRRE packaging DNA, 3.75 micrograms of PRSV Rev DNA, 10 micrograms of lentiviral expression vector,